Okay, and we're going to pick up where we left off before lunch. Go ahead and bring my screen back up. Is everyone seeing my screen okay? Is there anyone who can't see it?
Looks good.
Awesome. Okay. So before the break, we had just gone over some of the limitations of quantitative PCR. And so in order to account for some of these limitations, specifically the fact that there's a limited number of samples and a limited number of genes that you can evaluate at the same time, there was another technology that was born. Many of you guys have probably heard or maybe have even done some of this. This is microarray technology. So and how microarray works is we start with our samples of interest, in our case ground control samples and flight samples. We isolate the RNA, specifically mRNA, and then use reverse transcription to convert that mRNA into cDNA. And then in microarray, what we do is in our two samples, we label that cDNA with a fluorophore that's a different color. So in this example, we have ground control labeled with a green fluorophore, and we have our flight samples labeled with a red fluorophore. And then what we do is we have equal concentrations of the cDNA from both of our samples, and we expose it to a chip, and this chip contains a bunch of really tiny wells, and inside each of those wells is a small oligonucleotide, or small set of oligonucleotides, so ATCs and Gs that are complementary to known or annotated genes. And so depending on which chip you get, you can get like all of the genes in, say, the all immunology-based genes in a mouse or something like that. So you can, there's a variety of different chips you can choose from. You can create custom chips for specific sets of genes that you want to look at. But the idea is that each one of these little wells contains a set of oligonucleotides that are complementary to a specific gene in the perfect world. And so when we exposed our cDNA from our labeled cDNA from our two samples of interest onto the chip, what happens is that if that cDNA contains the gene of interest, that gene of interest will hybridize to its complementary probe and give off a signal. And so if a specific gene, let's say gene A, is expressed in ground control samples, what we would see is that those ground control samples would hybridize to gene A and produce a green signal in that well. If our flight samples express a gene, say gene B, then what we would see is all the cDNA from the flight samples, the complementary cDNA for that gene would bind to the probe and then produce a red signal. And then for genes that are expressed in both our ground control and our flight samples, they would both hybridize to the probes of, let's say, gene C, and then what we would see is a yellow signal being produced. So when we take a picture of our chip, or use it in a scanner to detect the wavelengths of light that are being emitted, what we ultimately see is a bunch of dots on a chip corresponding to expression of that specific gene in the ground control or flight samples or both. And these empty wells would be if that gene is not expressed in either sample. And so this is a picture of an actual microarray chip and what it looks like. So you can see here a combination of green dots and red dots and yellow dots. So again, in our example, anything in green is going to be expressed in ground control samples, but not in flight samples. Anything in red is going to be genes that are expressed in flight samples, but not ground control samples. And then these yellow dots means that they're expressed in both ground control and flight samples. And if it's that strong of yellow, it's roughly equal. If it's something like this that looks orange, then it's probably more expressed in flight than ground control, although still expressed in ground control. And so this gives us the ability to directly compare expression of flight and ground control samples for several, sometimes even multiple thousands of genes at the same time. But microarray comes with its own set of limitations as well. And so some limitations of microarray is similar to quantitative PCR. We can still only evaluate annotated genes. We can evaluate a lot more annotated genes at one time than we can with quantitative PCR, but they still all already have to be known genes, so no novel discoveries here. It requires hybridization, right? Our labeled cDNA has to hybridize to its complement on the probe, which always has the risk of nonspecific binding or the ability to cross-hybridize of sequences with high identity. So if some genes have a lot of similarities in their sequences, then they might not be different enough to prevent binding from to the wrong gene. So we could have some non-specific binding going on there. It's difficult to detect splice isoforms because, again, there's going to be a lot of similarities in the sequences of splice isoforms. The dynamic range is going to be limited by the scanner. So the scanner has to be set to detect a certain amount of photons when it's detecting the wavelengths. And so if you set the range of the scanner to detect a low amount, then your high expressed genes would be super saturated and you might not be able to tell subtle differences between higher expressed genes. Similarly, if you set your range of detection to more fluorophores, then it won't be as sensitive to those lower expressing genes. And even if you are designing or setting up your scanner to detect those low expressed genes, they're still very highly variable because you just don't get that much light coming from it since they're lowly expressed. It measures relative abundance, so absolute quantitation is difficult, right? So we're having two samples on here and just seeing based on the colors that they were labeled which one we're seeing more of, so it's relative abundance. There can be unequal, and there is unequal labeling efficiency of the fluorescent dyes. So labeling efficiency might be better with green than red, and then now you've introduced a bias into your system. There's chip-to-chip variation. So if you were to take the same two samples, put them on one chip, put them on another chip, it should be exactly the same. It might not be. So there has to be a background correction and normalization done to account for things like that. And then how you guys think of other limitations of microarray technology. Any questions about microarrays or PCR? Again, feel free to unmute your mic or put it in the chat.
Yeah, can I just go back to the last slide real quick?
For sure.
Thank you.
Thanks. Yep. You have a question?
Yeah, I didn't see the readings for the colors, the yellow present in both cells, GC cells, FLT cells. Okay, that's all I wanted to see.
Okay. That was easy.
Thank you.
No problem. Any other questions about microarray or PCR? Okay. So that brings us to RNA sequencing and why we're all here. So I'm not going to go through exactly all of prior RNA sequencing, so we'll just talk about the ones that exist, and we're focused a lot on next-gen sequencing since that's what we're all going to be analyzing. So before next-generation sequencing, back in the late 70s, is when Sanger sequencing came onto play. I know I was talking to Elba during lunch, who's had experience with Sanger sequencing and kind of knows limitations and how that can be difficult and challenging to work with and do. But Sanger sequencing is responsible for the completion of the first copy of the human genome. And actually, Sanger sequencing is still used today. So it's not a completely dead technology. But moving past Sanger sequencing, and I should mention that Sanger sequencing allows you to sequence short reads. And so if you were to want to put together or sequence an entire genome, such as the human genome, you would have to assemble those short reads together to put together a whole genome. So after Sanger sequencing in the 90s, next generation sequencing or pyrosequencing was developed, and we're going to get into this technique in the next slide. This is probably the most common form of sequencing used today. This also is very short sequences. Joel asked a question, should always Sanger sequence when presenting important data in good journals anyways? Why do you say that?
Sorry, I was just going to say, like, I've had this issue with a few people that I know that will do real-time PCR and a few other things, and RNA-seq or even proteomics, and you will still go back and saying a sequence what you thought your primers were, what your mutants were, whatever it is. You generally go back anyway.
Interesting. Okay.
And it's still the best quality, Joel. I mean, when it comes to 16S RNA sequencing for bacteria as well, getting a full length sequence, I mean, Sanger is still probably the best in doing that.
Agreed. I think it is still very useful. It's just everyone's moved to these high throughput approaches and they say that's where we're at. But if you're going to go ahead and confirm your high throughput approach, you need to step back and do Sanger in the end anyway.
Mhm.
You need an orthogonal means of backing your data up.
I don't entirely agree with this. Sequencing human genomes today are not done with Sanger, and we're not getting wrong base calls because of that. Long read technology
Yeah,
is fine.
microbes, not humans. I'm talking things of more steric nature. If you're doing microbiome work, you're working with a mutant of chlamydia or a mutant of Haemophilus or something like that.
Yeah, I think I agree with your point in general, but I think there's a lot of nuance here.
Yeah, I
We
think
could probably talk about it all day. Yes, agreed. Very much so.
definitely be highly debated for a long time. There are pros and cons to both, so I'll give you that. And again, I think that what Mike said in the chat here is it depends on what you're showing and what you're looking at. And I know I use Sanger sequencing to confirm when I order oligos for primers or something like that. I usually sequence them with Sanger to make sure that they are the nucleotides they say they are. So it is still used. Quite often, but the majority of sequencing nowadays is done with next generation sequencing. And again, that also being short reads, that if you were to want to sequence an entire genome, you would have to assemble those short reads. And we have begun third generation sequencing, which is single molecule sequencing through nanopores. And then this technology has been moving quite fast. Probably just over 10 years ago, it was insanely error prone. It was like 50% error rate, and kind of the short read or the short sequencing groups pretty much laughed at this long molecule sequencing. It turns out joke's on them, because nowadays nanopore technology can have as little as a 5% error rate or less. So it is getting better and better. And the advantage of sequencing an entire molecule all at once is that now you don't have to go back and assemble it. And so we are going to see more and more third generation sequencing happening. And in fact, there is a nanopore on the International Space Station, and we have sequenced microbes in space on the nanopore. So we are going to continue to see single molecule sequencing grow. I don't think that it's going to fully replace short read sequencing, just like next generation just didn't entirely replace Sanger sequencing. But I think we are going to see another shift where more and more sequencing is going to be on these nanopore technologies. However, our sequencing, the sequencing that we do in GeneLab, and still about 90% of the sequencing that's done worldwide is done through next generation or pyrosequencing. So we're going to get into the details of pyrosequencing, or more formally known as sequencing by synthesis. So in the pre-bootcamp survey, a lot of you guys told me that you're really comfortable with sequencing by synthesis. So if I'm going too slow, just let me know and I can speed up. So essentially what we have here is we have our sequence of interest here in red that we don't know the sequence of. So the goal is to find out what the sequence is of this red strand. And so what we do before we go into the sequencing by synthesis is we add some artificial oligonucleotides on either end in order to have a region that we do know the sequence of, so that way we can prime it, because of course we have to prime our sequence of interest in order to do the synthesis. And so they would have a primed region that we know about here. And then similar to PCR, what we would do is we add in our polymerase. But what we also do is, instead of adding in all 4 nucleotides, and the technology has moved past this, but I'm going with like kind of the original idea here. Originally, we would just introduce 1 nucleotide at a time, and that nucleotide would have a fluorophore attached to it. And so the idea is that 1 nucleotide would be introduced if that nucleotide was complementary to the next base pair in the strand that you're sequencing. It would form that hydrogen bond attachment here, and the phosphatase backbone as well, releasing a diphosphate molecule. That diphosphate molecule would then be catalyzed by the enzyme sulfurylase to form adenosine triphosphate, or ATP, the energy molecule. That energy molecule would then be utilized by luciferase, which catalyzes the cleavage of its substrate luciferin, which isn't depicted here, and luciferin gives off a signal, which is then a picture is then taken. And so basically all the places that have a signal, we would say, okay, a C is incorporated there. All the places that didn't have a signal, C is not the next base pair there. And then what's happened is a pyrase is introduced into the reaction, and that sequesters all the remaining cytosines in this example, and also the remaining ATP. And then a new nucleotide, labeled nucleotide triphosphate, is introduced, and this process starts all over again. And so that's how it goes about doing sequencing one base pair at a time. Of course, this takes a while. You can only introduce one nucleotide at a time. So the folks, Illumina is actually the company that really perfected pyrosequencing, or sequencing by synthesis. And how they made this faster and more robust initially is, instead of introducing one labeled nucleotide at a time, they labeled all 4 of their nucleotides, each with a different color fluorophore, so that way you can detect a wavelength that's going to be specific to one of the colors of the fluorophore, and then you'll know which base pair was introduced. They come up with even more clever ways of doing this as well, with actually even as little as just needing one color based on what they add to their nucleotides to be able to know if it is an ATC or a G. Any questions on pyrosequencing or sequencing by synthesis? We are going to go into this in a lot more detail. Any questions up front? Okay, great. So now just a broad overview of RNA sequencing. So we'll start with our samples of interest. In our case, samples from spaceflight or ground control samples. We would then isolate RNAs from those samples of interest. We then have to add on these adapters or linkers in order to, I should say we have to segment it, right? Because pyrosequencing is short read sequencing. And of course, when you isolate RNAs, transcripts can be several kilobases long. And so we want to chop those up, and then we add on these little linkers or adapters to allow them to attach to a flow cell, which is depicted here. And that flow cell has these little oligonucleotides that will be complementary to the adapters that we add, which we'll get into more in the next few slides, which will allow sequencing by synthesis to occur in each little area of this flow cell. You'll then be left with millions of sequences, and then you'll have to figure out where those sequences came from your respective model organism that you started with. And so you'd have to map them back on to a reference genome, and then quantitate how much of each one of the genes that are there in each of your samples, and you can perform differential expression analysis to then see which genes are up or down regulated, in our case in spaceflight compared to ground control. So that's just a brief overview. Now we're going to go into a lot of detail about this. So first, let's start with RNA. So as most of you guys know, and you can put this in the chat or just look at the slide, but what is the most abundant RNA in living organisms? Put it in the chat. Yeah, Mark, ribosomal. You can also see it depicted here. This is RNA, total RNA, taken from a universal human reference sample. And you'll notice that the bulk of it, over 90% of it, is ribosomal RNA. Of course, that makes sense. We're filled with ribosomes. Proteins are arguably one of the most important molecules that are made in living organisms. And so most of the RNA, the overwhelming majority of the RNA, is ribosomal. Ribosomal RNA is also incredibly well conserved and therefore not entirely interesting to us. And so in RNA sequencing, ribosomal RNA is often considered a contaminant. And so because we really want to focus and look at the more interesting RNAs, like our exonic RNAs, and sometimes even our intronic or our intragenic. So just a quick overview of RNA structure. This here is a pre-mRNA. And so as you guys are all very familiar with, we have the 5' untranslated region here and the 3' untranslated region on the 3' end. We also have a mix of exons depicted here in red and introns depicted here in this grayish blue color. And then that pre-mRNA is matured into mRNA by those introns being spliced out and those exons being brought together. and then finalize with this 5' cap being added on at the 5' end and a poly A tail being added on at the 3' end to make up our mature mRNA. And so mRNA can be very interesting. A lot of people, this might be that the primary thing that they're sequencing for is to look at messenger RNA, because messenger RNA, it will encode for proteins. And so if we want to see, if we're using an RNA analysis to assess gene expression, looking at mRNA is what you're going to want to do, and that's ultimately what we do. However, there's a lot more interesting RNAs besides just mRNA, and mRNA makes up a very small percentage of the total RNA sample, about 2 to 5%. But there's also intergenic and intronic RNAs, right? So there's could be long coding RNAs, there's Kiwi RNAs, there's small nuclear RNAs, there's microRNAs, of course, so all sorts of interesting RNAs that we might want to recover in addition to just mRNA. But if you are just interested in mRNA, you can use a technique called poly A enrichment, so taking advantage of this poly A tail on the 3' end of mRNA and using that to positively select for mRNA in your sample. When you do that and then you assess the different RNA types in your sample, what you'll notice is now the majority of it is exonic RNA, which makes sense, right? Most of this is made up of exons here. However, we do also see some intronic and intergenic RNA. And important to note, you see the percent of ribosomal RNA drastically reduced when you do positive selection with poly A. I do want to ask you guys the question, though, if we are positively selecting for this poly A tail, which should only be on mRNA, why do you think that we're also getting intronic and intergenic RNA using this approach? You guys can unmute your mics, type it in the chat. Any guesses? Alternative splicing. What specifically about the sequences, though, do you think would cause us to recover some intronic or intergenic RNAs? Repetitive regions? Yeah, for sure. So when we're positively selecting for this poly A tail, it's a string of A's, right? So it's a string of adenosine molecules. And so we would have an oligonucleotide that is a string of T's. And so those T's would be complementary to poly A tails, right? And then we could, attach those string of T's to a magnet and then pull down our everything that has a poly A. However, we're just looking for a string of A's, right? So as Ty pointed out in the chat, other types of RNAs, like intronic and intergenic, have strings of A's within them. And so although we're specifically pulling out strings of A's, because strings of A's can also exist in intronic and intergenic RNAs, we're inadvertently going to be pulling those down as well. So we're inadvertently going to be having those in our samples as well. And of course, this is, if you're going to do the poly A selection, you really don't want to put much weight on your intronic or intergenic regions, because those were selected in a biased manner, right? So the only intronic and intergenic regions you're going to have are those that have poly A's within their sequence. And so when you do a poly A positive selection to enrich for mRNA, mRNA is pretty much what you want to look at in your samples, and you can't really do much more. So because of that, and because GeneLab really wants to accommodate and provide as much information that would be useful to our users as possible, our standard is to do ribodepletion. So rather than positively select for a poly A tail for messenger RNA, you can also negatively pull out ribosomal RNA. So rather than designing an oligo to identify poly A tails, you could design oligos that are specific to the sequences of ribosomal RNA. And then instead, in your sample doing a positive selection, you could do a negative selection in which you pull out and remove all of your ribosomal RNA. And now when you do that, you should be left with, as you see here, a mix of all the other different types of RNAs that exist. And hopefully, if your ribosomal depletion method went well, you'll see very little ribosomal RNA in your sample. And so that's the standard for GeneLab. However, There are reasons, and like I said, if you're only interested in differential gene expression, then you might want to go with mRNA. And in that case, one reason why people would do that is because you don't have to sequence quite as deep, because most of your sample is your exonic regions. So it could save some money if that's all that you want to do with your sequencing. But if you're interested in more things, like many of our users on GeneLab are, you'll definitely want to go with the ribosomal depletion. Caveat of that being, if you do want to use that information to do differential expression, then you do want to sequence a little deeper, as we'll get into a little later. But so these are the different ways to start preparing your total RNA and getting it ready for selecting what you want to do sequencing. So now that we've learned how to clean our total RNA for what we want, just a quick overview of Illumina's sequencing workflow. And I say Illumina because Illumina is an incredibly successful biotech company. It specializes in next generation technology that uses the pyrosequencing method. And it is so successful that about 90% of all sequencing worldwide is performed on an Illumina instrument, including at GeneLab. So at GeneLab, we do have a NovaSeq Illumina sequencer, which we use to sequence samples in-house. That said, as we discussed earlier, we know that nanopore technology is continuing to gain momentum and popularity, and with ever reducing error rates in that technology, we probably are going to see a shift. However, I don't think that short read sequencing will become obsolete, but we will probably see less of that global reign on the sequencing technology held by Illumina. So the Illumina sequencing workflow has the following 4 steps. The first would be library construction, cluster formation, actually sequencing your regions of interest, and data processing and analysis, which we're all going to do together. So I do want to go into detail about library preparation because a common phrase that's used in bioinformatics is crap in, crap out, meaning that if you don't prepare your samples properly, then there's only so much we can do on the in silico side to correct for mistakes that were made on the wet lab side. So it's really important that the library preparation be done correctly and that you put high quality libraries into the sequencer to make sure that you get high quality data at the end of the day. And so the first step in library preparation for us, so I'm going to go over the ribodepletion method since that's what we use at GeneLab. And so it's going to be a negative selection method, very similar for poly A. It's just this first step here, instead of depleting the ribosomal RNA, you're actually going to enrich for the poly A. So we're going to talk about ribodeplete. And so in here, we start with our isolated total RNA from our samples of interest. And the first thing we're going to do is remove ribosomal RNA. So how we do it with the Illumina system is we use biotinylated target-specific oligos that are specifically targeting ribosomal RNA, and the biotinylation will allow it to be magnetic, along with ribosomal RNA removal beads. So we'll be able to pull down based on that biotinylated oligonucleotide that's complementary to ribosomal RNA. The complement will attach to any ribosomal RNA in your sample, and then we'll be able to use a magnet to pull down that ribosomal RNA, and we kind of keep the solution and remove our pellet, which contains just the ribosomal RNA. After we've removed ribosomal RNA from our samples using this method, we then have to, got ahead of myself, we then have to cleave that RNA. So again, we're doing short read sequences, and so we need the RNA that we're building for library preparation to be short. And how short depends on how long you're going to sequence. And so Illumina sequencer, I think the most they can sequence is 2 by 250. So 500 cycles or base pair in total. If you do that paired end, 250 by 250. That's usually done in like metagenomics type of analyses. For most analyses in the RNAseq world, usually 150 is a pretty good length, and that's actually our standard in GeneLab. So we sequence it paired end 150, and so that means that we are going to sequence 150 base pairs in the forward direction and then 150 base pairs in the reverse direction. And so because of that, we want these fragments, since we're going to sequence 150 base pairs and another 150 base pairs in the opposite direction, we want that fragment to be anywhere from 250 to 300 base pairs is kind of what we're shooting for. I'm too young to know this, but can I ask people who have used Sanger sequencing before how much harder it was compared to Illumina? I don't know, Elba, if you're online, if you want to respond to Chief's question. Actually, I think that she mentioned that she might be a little late. I'm gonna let Elba talk to you about that, 'cause she has a lot of experience with it. I've never actually done it myself, so I only know stories from my professors that have told me how spoiled we are. Those slab shell sequencer is a pain. Yeah, there's also Elba put in the chat a good link to learn more about Sanger sequencing. So I'll put that in here. And then I also, excuse me for a second, have another link too that goes more in depth on Sanger sequencing. So this was done on a gel. It used a, the sequencers were actually cut off at certain base pairs. So they used modified illegal nucleotides that'll actually prevent any subsequent sequencing beyond that nucleotide once it's incorporated. And then you run it on a gel, and it's usually an lithium bromide gel, and so you have to expose it to UV to see your bands. I don't know if you guys have ever used lithium bromide to detect DNA on polyacrylamide gels. I've gotten sunburns because of it, cutting out bands on gels. It's a lot easier doing Illumina sequencing.
So I have this hanging up in my office, and this is really old school sequence data. That so much more difficult to interpret than current data.
Wow, that's fascinating. How old would you say this data is? 1960s? 1970s?
No. It doesn't have a date on it. It's sometime in the 90s.
That's really cool. I've never seen a Sanger sequencing chart before.
I mean, it's a little different. This is, modern Sanger sequencing uses the fluorophores, and these are radio labeled. So it's definitely different.
Yeah, and as Wygen pointed in the chat, that Sanger sequencing now, you can get it done for less than about $10 per sequence, and they use a capillary gel, so a lot less starting material, which is a bit more convenient using the fluorophores as well. So they have improved their technology on the Sanger sequencing that is used still today.
So it's still much cheaper than Illumina then? Or no, they're about the same price?
Illumina is going to be cheaper.
Oh, okay, I see.
Mm-hmm. Yeah. Illumina, but Illumina, you want to do like a lot of sequencing because it's a high throughput. So if you only want to sequence, like for instance, I used it when I was a postdoc to sequence illegal nucleotides that I made for primers when I was doing qPCR reactions. So I would order in an oligo a primer, and then I'd want to confirm that it was indeed the sequence that I ordered. And so I would send it off for Sanger sequencing. So when you want to just have little bits of pieces to sequence, it happens.
Yeah. Even on Illumina, the prices can vary. My lab's doing a MySeq run next week with some 16S data, and that's going to cost us about $1,500. We're also doing a big metagenome project, and that's going to cost us about $20,000. So, the answer is it's a big depends.
Yeah.
Price per base is certainly cheaper though.
Right. Yeah. So price per base. So if you're sequencing like a ton of stuff, like Rachel was just describing with her big metagenome project, it's going to be cheaper per base to go with Illumina. But if you're just sequencing, short little sequencing, then you can get away with doing Sanger. Okay, so once we've removed our ribosomal RNA and chopped up our remaining RNA into short fragments, again, depending on how long you're going to sequence, what your read length is. So as I mentioned, in GeneLab, we do paired N150, so 150 in one direction, 150 base pairs in the other direction. So we're looking for these sizes to be around 250 to 300. When we do that of course has to be optimized because what in the kit that Illumina uses that I'm going over with here, what happens is it uses, I think it uses divalent cations that it uses to do the random chopping, because you ideally don't want your chopping up of your RNA to be biased. So you want to do something that's unbiased. So divalent cations offers a way to do that. And then once you have your RNA that's ribodepleted and now all nice and chopped to the size that you want to go ahead and convert your RNA into cDNA, because we only sequence cDNA, or we only sequence DNA. So the first thing you want to do is synthesize your first strand of cDNA. So very similar, again, really cool thing about sequencing, it kind of takes advantage of all the technologies that came before it that I went over. So this takes advantage of using reverse transcriptase to convert our urisols to thymines. And so we have our first strand cDNA synthesis from our short RNAs that we have now after doing the chopping. The next thing that we want to do is synthesize our second strand of cDNA. So after you do the first strand synthesis with reverse transcription, you're then going to do your second strand cDNA, and we actually use a stranded kit. So one thing that I haven't mentioned is RNA can be transcribed from the sense or the antisense DNA strand. So coding RNA or message RNA is going to be transcribed from the antisense DNA strand in order to make a sense message RNA, and that can be coded. However, non-coding RNAs can be transcribed from the sense DNA, making an antisense RNA. And because there's so much repeats in genomes, like Ty mentioned in the chat, there is a chance that you'll have part of your sequence. Remember, we chopped these up. So there is a chance that you'll have part of these sequences that match up with a transcript that was in the sense direction and also matches up with a transcript that was in the antisense direction. So if you don't keep track of the strandedness of your RNA that you start with, you will now not be able to know when you go back to try to identify where these sequences came from, you won't be able to know if it came from the sense or the antisense DNA strand. And so maintaining strand specificity is going to be important. And how Illumina does this is when it synthesizes its second strand of cDNA, it uses a polymerase that will incorporate urisols instead of thymines. So that second strand is going to have U's in it. And this is going to be important in the last step of library prep to maintain the strandedness. And the fourth step in library preparation is to add on these add on these A's, so these adenosines here, to the 3' end of your cDNA. This one provides a nice overhang for us to add our adapters onto in the next step. The other thing is it will prevent these from sticking onto each other. And so in our next step, what we want to do is we want to add on our adapters. So keep in mind, this is our sequence of interest, right? This is what we want to sequence. But right now, we don't know what that sequence is. This is just a short piece of cDNA that we derive from our samples. We currently don't know what the sequence of it is. The goal is to find out. So because we don't know what the sequence is, and we're doing a method called sequencing by synthesis, as I showed you guys when going over the PCR reactions, in order to synthesize a new strand, we first have to prime it in order to get the polymerase to latch on to be able to synthesize a strand. And so if we don't know the sequence how can we prime it? Well, what we do is we utilize these A's, this overhang that we just added, and then we add on sequences that we do know. So these are called adapters. Our adapters have a specific structure in Illumina sequencing. So there's a P5 side and a P7 side. And this sequence here, indicated in green and in red, is going to be complementary to our flow cell. And we'll get into that a little bit more when I show you sequencing. So these oligos, we know these oligos, we know what the sequences are. They're going to be complementary to the flow cell. We also have in here this molecule in blue. This is known as an index. So what an index does is when we do sequencing, we're going to combine short pieces of cDNA from multiple samples, right? Because this is high throughput, so we can sequence a lot at once. And so if we're going to mix all of these sequences together from different samples, we have to have a way to be able to identify which sequence belonged to which sample after we do the sequencing. And so how we do that is in these adapters, in addition to having a region that can attach to the flow cell, we also have a short piece of oligonucleotides that we're going to add to every single piece of cDNA that came from the same sample. And so every piece of cDNA that came from one sample will have one specific index. If it came from another sample, it'll have a different specific index. And so the uniqueness of those indices will then allow us to know, okay, this sequence has this index, so I know it came from this sample because I'm the one that controlled what sample received that index. So we add that on. The other part, and I just want to point out, this is called single index adapter because you see that there's just an index on the P7 side. So I'm going to get in a little bit more into different types of adapters in a second, but keep in mind this is single index adapters. The other part of this adapter is going to be a priming region. So this is going to be the region that we are going to artificially add on to our sequences of interest in order to know this sequence. So that way when we do the sequencing by synthesis, we design a primer. We don't, a company does, Illumina in this case. We design a primer that's going to be complementary to this priming region here. So that will allow the priming region to latch on, which will allow the polymerase to latch on and then to do the sequencing. And so how we add these adapters on, we're going to add them on to either end of our cDNA, is they also contain this T overhang. So if you remember the last step that we went over in the previous slide, we added on that A to the 3' end of our sequences of interest. And so now these adapters have this T on the 5' end of the adapters. And so we then mix this with the enzyme ligase, which will attach strands of DNA together. And so this T is going to be hybridized or ligated to this A on either side of our cDNA molecule. And that's how we are going to get our adapters added on to our sequence of interest. And so now that we have our adapters onto our sequence of interest, before I get into the next step, I do want to go over a couple of different types of indexes, or I should say adapters. So the one that I'm showing here is a single index adapter. So you'll see by single index, I mean we just have an index on the P7 side of the adapter. We do not have an index on the P5 side. So this used to be a very common way to do sequencing, but what can happen during the sequencing process is kind of fragments of the same index that should be latched onto a sample might be left behind, might come unattached from your region of interest, and then another region of interest from a different sample might latch on to this index that was allocated for a different sample, and that meant that kind of issue is known as index hopping. And that was a bit of a problem in earlier sequencing in Illumina days. And so one way around that is to put an index on both sides. So now the likelihood that both of these indexes are going to hop and both hop to the same sample that's different from the one that it was intended to is really reduced when you have one on both sides. So this is called unique dual indexing. Again, dual because there's one on the P5 side now, as well as that one on the P7 side. So now every sample has two different unique sequences to identify that this sequence of interest came from a sample with a specific I5 and a specific I7 index. The other thing that we can do, we'll get into this in a few slides down the road, another issue that can happen is in the library preparation, the very last step that we're going to go over is an amplification step using PCR. And so when we do that amplification step, we do it to make sure that there's enough material to be able to sequence. However, it introduces artificial duplicates, right? So we call those technical duplicates. And artificial meaning that there was only a copy of it because we artificially made a copy of it in the last step of the library prep, which is to amplify. Of course, biological duplicates can happen, right? So if you have a really highly expressed transcript, and although we do the divalent cation to chop up our transcripts, which should be unbiased, there's always going to be some level of bias that happen. So if we have a really highly expressed transcript and we introduce the divalent cations to chop it up, there is a probability that those divalent cations will chop up this multiple copies of the transcript in the same place, giving you the same exact sequence in your insert. So that would be a biological duplicate, right? Because it came from biology because of the nature of it being highly expressed and using the divalent cation chopping up. And so in order to distinguish, once you get your sequences back, if you have a set of sequences that are exactly the same, in order to distinguish if those sequences were technical duplicates or true biological duplicates, what we can do is add this UMI flag here on the index. And this UMI is a string of 9 to 15 base pairs long that are completely random. And so you're going to have a unique, well, the goal is as long as you have enough base pairs. So I would recommend 10 or 11. We actually only have 9, so there's only like 300,000, I think, unique combinations. But still, really low likelihood that the same UMI would be associated with the same exact sequence. So the idea here is this is completely random. And so when we add this on here, now we know this is before we're going to do that last step, which is the final amplification of the library. And so we'll add these onto our sequences. So now when we go and we get our sequence back, if we have 2 sequences that are identical and they have a different UMI, then they must have been a biological duplicate and a true duplicate. If, however, we have sequences that are identical and the UMIs are also identical, since we added on these UMIs before our amplification step, that means that when we amplified this library, we also amplified that UMI, which is why it's the same. So that would indicate a technical duplicate. And so that helps identify and ultimately, debatable again, remove those artificial or technical duplicates. So a few different ways a few different adapters to choose from. On GeneLab, our standard is to do the dual index UMI adapter. So we have an index on both sides, the I5 side and the I7 side, and then we also have this UMI to be able to identify technical duplicates. Our next step in library preparation is that final step that I kept referring to in my previous slide, which is enriching the DNA fragments. So a couple of things about this step. Again, we want to enrich these fragments to make sure that we have enough to be able to sequence. The other thing that we do here is we take advantage of the previous step in library preparation when we did that second strand, cDNA synthesis, where we synthesize with the polymerase to incorporate urosols instead of thymines. Now when we do this enrichment step, what we do is we add in a polymerase that will not synthesize past a uracil. So once this polymerase hits a uracil, it'll fall off and it'll stop working and it won't make any copies. This is important because now the only strand that we're copying is in one direction. We're only copying now that first strand. So this is how strandedness is maintained in Illumina's stranded library prep kit. And then once we do our amplification step, we now have our final libraries that are going to be ready for sequencing. So this is our final library. We have our P5 end on the 5' end, our P7 end on the 3' end. Again, this example is single index, so we only have this index, which is used to identify which sample the sequence came from. We only have it here on the P7 because it's a single index adapter. And then we have our priming regions, right, from our adapters, and then we have our sequence of interest. And of course, these were all derived now from the same strand, so that way we can maintain strandedness. That is the end of library prep. I am going to go through how we QC our libraries, which is incredibly important before sequencing them. But before I do that, is there any questions on library preparation? Any of the steps I just went over?
Thanks for reminding us that magic is real.
I do have a quick. Oh, sorry, I do have a quick question. So, how many unique indexes are there? So, when they do a single cell RNA-seq, I assume then sample from each cell has a unique index or barcode. So, what's the limitation on the design there?
Yeah, so I mean, it's up to your imagination. We can synthesize nucleotides. I can't remember how long. We can 3D print oligos now. There's a couple companies, at least in San Diego, that do that. But yeah, so it can be as long or as short as you want. The goal is we usually get it in 96 well plates, and we get it, our oligos that we use are 6 for the indexes. So for this part here are 6 base pairs long, and we have 96 of them, right? And the goal is to make sure, right, so there is going to be some errors in sequencing, so we want it to withstand at least 1 base pair mismatch. That means that each one of our indexes has to be different by at least 2 base pairs, right? So that way, every single index, because you need these to be unique for each sample. So if we have a 96 well plate, we have a 6 base pair long index. Each one of those 96 indices are going to differ by at least 2 base pairs. So that way, if there's an error in the sequencing when you sequence the index, it can withstand 1 error, and you'll still be able to identify it to which sample that it came from. In terms of single cell, so single cell adds a couple of additional barcodes. So single cells all use UMIs to identify and remove duplicates. It uses indexes to identify samples, and it also uses a cell ID barcode, right? So in addition to knowing which sample your sequence came from, you also want to know which cell from each sample it came from. So it has an additional barcode known as a cell barcode. And because there's so many oligos that are just barcode, it's actually one whole sequence. So you sequence single cell and paired end, and one of those sequences is just barcode information, and then the other sequence is your sequence of interest.
Okay, so would your insert sequence have to be much shorter to accommodate all those extra links from what's added?
So that's what I'm saying. So yeah. Yes and no. What happens is, so yes, it is usually shorter. So we usually sequence, I don't know if Lauren's in a meeting. She's our single cell expert, but it's usually just under 100 base pairs. And then there's about 30 to 40 base pairs that are just cell ID, cell barcode, UMI, and index. And so we sequence in both directions in that case, right? So we have one strand, right, as an example. Of course, everything's double-stranded. But on that strand, one side of it is all barcode that was added on, and then the other side of it is your sequence of interest. So when you sequence in both directions, a lot of these, and it depends on what kit you're using, but you'll have your forward read is going to be only sequencing the barcode. And so you can set your sequencer to sequence a certain number of base pairs. And so you know how long your cell ID is, you know how long your index is, you know how long your UMI is. And so what you do is you tell Illumina to sequence the exact number of base pairs of all of those barcodes in the forward direction, because that's the side for at least 10x kit that your barcodes are on. And then your other side, which contains your sequence of interest that all these barcodes were attached to, you sequence, let's say, up to 100 base pairs. And again, you can increase that. It depends on how big you made your Cdna fragments when you did your chopping phase. And it also depends on the capability of the sequencing instrument you use. So I actually always get frustrated because it's just become the standard to sequence around 35 to 40, depending on how big, sometimes longer, all that barcode information is. And then they usually sequence like less than, it's like around 92. It's a random number that they sequence in the opposite direction, which contains your sequence of interest. But in reality, in a sequencing machine, if you have like a paired N150 capability, you can set it to sequence 50 base pairs in one direction to get all of your barcode information. And then you could sequence presumably up to 250 base pairs in the opposite direction for a total of 300 and get a lot longer reads. I don't know why people don't do this. I've asked about it. Our project scientist kind of like makes those final decisions for our standards. And just because it's like industry standard that people do it shorter, I'm actually not entirely sure why. I don't know if anyone does single cell, if they have a better reason why we don't sequence longer into the region of interest. I'd love to hear it, because I haven't found a satisfactory answer other than this is the way people do it. And this is the way people do it is not a justification to do anything in my mind. So.
Okay, cool. Thanks for answering my question.
For sure. And I'll let Lauren talk a little bit more about single cell too when she joins us again. Any other questions about library prep before we move on to Okay. So let's go on to quality control. So we do want to make sure that our libraries are of high quality. Because remember what I said, crap in, crap out. We don't want to put poor quality libraries into a sequencer because we're not going to be confident in the interpretation of the sequences when they come out the other end. So we do a QC on our libraries. We do this using electrophoresis. It seems like just from our conversations, a lot of you guys are familiar with electrophoresis, but just to go over it quickly. So we have a polyacrylamide gel here within a buffer, and then you load your samples of interest into each well in addition to a marker. I think our marker is, it looks like our marker is in both the front and the end well. And so that marker is going to be chopped DNA of known sizes. So that way, you know, if you have chopped DNA here, our smaller fragments are going to be at the bottom, our larger fragments are going to be at the top. And the reason is because after we load all of our samples in each well, we apply current to this buffer, and we have electrodes. Our negative electrode is going to be at the top where the wells are. Our positive electrode is going to be at the bottom. Anyone know why that is?
Because it'll run the DNA downwards towards the positive?
Yeah.
DNA charge.
Exactly, DNA charge, yeah. So DNA is negatively charged, and so it will go away from the negative electrode and toward the positive electrode. For those of you guys who do gel work, I have done this more times than I care to admit. If you plug in your electrodes in the wrong order, you will lose your sample because it goes up and off the gel the other direction.
It's a badge of honor, Amanda.
It's really frustrating. So I got used to whenever I'd start a gel, after that happened the second time, I would sit there and wait to watch it run a little bit. So that way, if I saw it wasn't going the right direction, I'd quickly stop and turn it around. And so this allows you to know the size of your fragments. And of course, remember, we're chopping up our libraries to be a certain size based on how long we want our sequences to be. And so this will help us with the size check. We also know how big ribosomal RNA is. So this is also going to help us check for ribosomal RNA and if we actually removed our ribosomal RNA. The other thing this is going to check for, which is insanely important, is adapter dimers. So those adapters, they can come together and form an adapter dimer, which we're going to get into in quite a bit of detail. And that is horrible for sequencing. It is a really tragic contamination that you definitely want to get rid of before you sequence. And I will show you why in the next few slides. But first, taking a look at this QC. For any of you guys who have never seen an electropherogram, this is what it looks like. And so an electropherogram is basically just taking the position of these bands and putting it in a graph format. This is one that looks at bands coming off over time. So the longer it takes a band to be resolved off of a gel, the larger it is, right? It's going to take a longer time to go through the polyacrylamide. It's a larger fragment. Smaller fragments will come off a lot sooner. And this was done with fluorophore, so fluorescently labeled RNA in this case. And what we see, this is an electropherogram of total RNA. So as you can see, the overwhelming peaks that we see in this electropherogram are, of course, going to be from our ribosomal RNA. So this is total RNA before any depletion. And you see this is human because we have 28S fragment here, an 18S fragment, and then your 5S region. And then this here is just a marker to know when your peaks should start to come off. And so this is what we want to make sure is gone when we run electrophoresis on our libraries. And so for an RNA library, one that looks good, you could run this. This is a small gel depicted here. And then over here is similarly these peaks graphed, except this time it's graphed where base pairs are here on your x-axis. So it kind of converts the time. Thanks to your markers, you know how long things you know what size something is based on when they come off because you have markers that tell you that. And so then you can convert this time into base pairs. And so you see here that this RNA library, we see a nice peak here right around the 300 to 400 base pair region. So that's pretty nice library. And this is what it looks like here on the gel. So we got our heavy marker up top, our smaller marker on the bottom here, And then we have this smear here that's our library. And of course, remember, we are just chopping up the library. I actually don't think I mentioned, which is actually really important. So the last step of our library preparation is actually a filtering step. We actually do it before the PCR amplification. So I should say the time before. So after we add on our adapters to our sequence of interest, we do a size selection filter to remove any of the nucleotides we had, the ligases to add on, and also to remove ideally any adapter dimers. So we do a size selection to make sure that we're removing all that stuff before our final amplification. And so here we're looking to make sure that we don't have any of those smaller peaks, which would be adapter dimers, which I'm going to get into more of. Of course, when you have a ton of samples, a ton of libraries that you prepared for sequencing, you're not going to want to sit here and load gels all the time. So there's fancy micro gels that are done called bioanalyzers or tape stations, and that's what we use at GeneLab, a bioanalyzer, and that creates an electropherogram to assess quality, so you don't actually have to read the gel yourself. It's all done on a nice little kit, and then you put it into a computer, and it makes little the kit makes these little micro gels, and then it's read by the computer to see the size of your libraries. And so here's an example of a couple of libraries that were prepared for sequencing. These are both examples of pretty good libraries. So this is kind of what you're looking for here. So we see we have our initial lightweight marker over here on the left. Again, this x-axis is in base pairs. And then we have our higher, heavier marker here on the right. And so we want to see, again, depending on how much you're sequencing, I think I asked this question, so I might not want to yet. Yeah, I do. So we can tell the size of our library, and before I get into reminding myself that we filter, before we go into that last step, I was saying that the reason that this is not just one nice straight peak and instead a smear, it's because during the step where we chop up our RNA into small pieces, we can't exactly control how big they are. We do size selection, and so based on our size selection that we do, they're going to be within a range. And so that's why we see kind of a range of sizes here. Notice that this is pretty much the only peak we see, of course, in addition to these markers. That's what we're looking for. We don't want to see any other noise on either side. You see that you don't see anything big that would represent ribosomal RNA in here. We're really just seeing the library. So this is what we're looking for. This is another really good library here. Probably the biggest difference between this library and this library is slightly different in the range of sizes, and there's more size variability. The goal is to try to get this as tight as possible to all the same insert sizes, because there are biases when you do the sequencing based on size, which we're going to get into. So I would say that this top library is probably a little bit nicer than this one down here, but they're both pretty good. Importantly, there is no peak around here in between your light marker and what you expect your library size to be. If you do see a peak that is around 100, so adapters are depending again on if you have dual index adapters, if you have UMIs or not. Our adapters can be around 75-ish base pairs. So when you're looking at an adapter dimer, which are two adapters that have come together to make an adapter dimer, you're looking at about 150, again, plus or minus depending on the nature of your adapters. And so you do not want to see a peak at around 150 base pairs, because that indicates adapter dimer contamination, which is a horrible thing to have in your libraries. And so just a summary here of when assessing library quality, we want to look for the following: no adapter dimers. So before I tell you the answer, I'm curious, any guesses why we wouldn't want adapter dimers in our libraries? Yeah, Joel, because then, yeah, Susie, yeah, because then we would just be sequencing our adapters. We already know those sequences, so that's a huge waste of money if all we're doing is sequencing adapters that we already know the sequence of. So we really want to make sure those are away, and there's even a bigger problem with this contamination too, which I'll get into. We're also checking to make sure that library size is consistent with the number of desired sequencing cycles. So again, your sequencing cycles are going to be representative of the length of the sequences that you want. And so if we're doing, for example, paired end sequencing at 150 base pairs in each direction, we want ideally our inserts to be 250 to 300 base pairs long. And then you have to remember, this is the whole library that we're looking at. So our sequence of interest, which again, for paired end 150, we want to see around 250 to 300 base pairs. But then when you're looking at these gels, you also want to account for the adapters that you added onto either end. So that'll be an additional roughly 150 base pairs. So really between the 400 to 450 base pair range for the sequencing that we do would be ideal. And I kind of probably gave this answer already, but let's talk about it. So I was doing PE 150. So what if you're sequencing at paired end 100? What is a good library size? Keep in mind that if you're doing, that your adapters will be about 150 total, 75 for each end. So if you're sequencing at paired end 100, what would be a good library size? Keeping in mind that you also have to include the size of the adapters. You can go ahead and put your answers in the chat. No guesses? So we're sequencing in this example, paired end 100, kind of 300. Like it? Any other guesses? 200 to 250, okay. 150, 250. Okay, I'll post the same. So let me go back a couple slides. Sorry, my computer kind of hides the corner of my monitor. I'm gonna go back a couple slides. Okay, so we have our final libraries here. And we know that each of these adapters, so this library is what we're running the electrophoresis gel on, right? And so we know that we want to sequence at paired end 100. So if we want to sequence this insert at paired end 100, paired end means we're going to sequence 100 base pairs in this direction and then 100 base pairs in this direction. That means that the total insert that we want to sequence should be around 100 plus 100, right? So the insert we want to have is around 200. But then we also have to account, because we're running this electropherogram on the full library, which means we've already added our adapters. So we want to see at least 100 in both directions, so 200 base pairs making up our sequence of interest, and then roughly 75 base pairs for one adapter and roughly 75 base pairs for another adapter. So that's going to be 200 plus 75 plus 75, so roughly 350. So 300 to 350 would be the range that we would want to see our libraries if we are sequencing paired end 100. Does that make sense? Is there anyone that wants me to go over how I got that again? Yes, makes sense, or yes, you want me to go over it again?
So essentially it'd be the 100 in each way, plus 75 plus 75 times 2.
Exactly. Yep.
Is that for like any size, or is that dependent on the amount of base pairs with the starting PE?
It depending on the amount of base pairs that you plan to sequence. So you kind of want it right, because when we're chopping up our RNA, way at the beginning, so way at the beginning of the library, after we remove our ribosomal RNA, and then we're starting to chop up our RNA of interest, when we're at this step, we already want to know what we're sequencing at, because this is going to dictate how long we should do this digestion for. So if we're sequencing at a shorter length, then we would want to digest this longer, right? Because the longer you digest, the shorter your fragments are going to get. If we're sequencing at a longer length, then we want to digest this for less amount of time, because the less amount of time is with the divalent cations, which are doing the chopping, the larger the pieces are going to be. And then we will size select too to make sure we're within the range that we hope to be. Yeah, so it
Okay.
depends how long you
Okay.
want to sequence. Yeah. So it's
That
really
makes sense.
important when you guys are designing RNA-seq studies, talk to your bioinformaticians before you do your experiment. Incredibly important because it'll dictate. Go ahead.
Sorry, just one last question.
Sure.
So I usually get stuck in the weeds when it comes to details and variables. So like we started with 100. Let's say if it was 50, would it be 2 50s and then 2 75s?
Exactly.
Okay, so the 75s are constant.
Not entirely constant, so it depends on the nature of your adapters. So,
Okay.
and I know, guys, we're into our break. I'm just going to finish answering this question. So if you guys don't want to hear the answer, feel free to take your break now. But yeah, Mark, so it depends on what your adapters look like. So remember I went over these 3 different kinds of adapters for bulk RNA sequencing. So depending on how many base pairs your index is, depending on if you have 2 indexes, right? So if you have an index on either side, so let's say you have 6 base pairs for your index, and if you have 2 indices, now that's 12 base pairs, plus the size of your P5 and P7 region, plus the size of your priming regions. Or you might want, like GeneLab does, have dual index with UMI. So then you also have to account for the size of your UMIs. So that it's like roughly 75 total, but that's plus or minus depending on the nature of the index that you're using. That make sense?
Yes, it does actually.
Cool. Okay, guys.
So,
go ahead.
I'm sorry, go ahead.
Oh, I was just going to say we are into a break. I'm happy to stay on and continue answering your questions, but if anybody else wants to go ahead and take their break, please go ahead and take your break, and we'll be back here by... Wait, are we at the break time yet? Sorry, let me see the schedule. Just hang on to your question real quick. I couldn't remember if it was 1:10 or... Oh no, we're good till 1:30. Yeah, sorry guys. We have 20 minutes before the break. I hope no one left. Sorry, Mark, go ahead and ask your other question.
Oh, it was more like I just try to like get it in my head. Like a good mental equation for me would be like the PE number times 2 plus adapter number time or adapter length times 2.
Exactly.
Okay, that's cool. Thank you. That actually really cleared it up for me. I'm gonna write that down right now.
Are the adapters always the same size on both sides, Amanda?
Oh, I mean, you get to make them up. So went too far. So not necessarily. Let me back. So one more slide, guys. One more slide. Okay. So for instance, your adapters here, right? So in your single index, the adapter on your P5 side, because it doesn't have an index, is going to be shorter by the length of the index compared with your size of your P7 adapter. Yeah. So they will be the same size if you have a dual index and each index is the same number of base pairs, which is common when you do dual index. So like for a dual index, your P5 adapter and your P7 adapter are going to be the same size. If you have, like we do, dual index UMI, your P5 adapter is going to be fewer base pairs than your P7 adapter if your P7 adapter has the UMI on it. So I know I did just tell Mark times 2. Again, that's a rough estimate. So they will be roughly equal because the bulk of the sequence is made up with the P5 oligo, the priming oligo. It's going to make up the bulk of it. But then, right, because indices are like 6 base pairs. So they might be different plus or minus 6 base pairs, or UMI can be up to like 12-ish base pairs. So they could be different by 6 to 12 base pairs.
Right, and we're just coming up with a rough area anyway, because
Exactly.
the digestion is not a hard science.
Yeah.
I follow.
No, but that was a good point. So Mark, I know I said 2 times for the adapter length, but Mike's right. Your adapters can differ by 6 to 12 base pairs, or right around there, depending on the size. Yeah. Any other questions about library prep or quality before I continue to nail down the adapter dimer issue?
Amanda, I have a Did you question. address RNA quality?
I did not. But that's a very good
Because this is
point.
only as good as the RNA you start with.
Yeah, so great
So are
question.
there any parameters we should be aware of, like with the RIN and all of that? Because
Yeah.
we basically, if it's not at least a 9, we prefer 9.5. We don't do it, but I've seen people do a lot less. So it would be helpful to talk about how that affects what we get.
Yeah, so your RIN value is a measure of your RNA quality. However, it essentially, and if someone knows this better than me, please chime in. How I think about it is it essentially is assessing how intact your RNA is, right? So if you have highly degraded RNA, your RNA is going to start off being shorter, being fragmented, right? Whereas really high quality RNA is like intact RNA. So your RNA starting fragments when you isolate your RNA are going to be larger, right? So if you have intact RNA, you're going to have a higher RIN value. If you have degraded RNA, you're going to have a lower RIN value. That said, one of the steps, the very first step in library preparation, well, technically the second step after you remove ribosomal RNA or select for poly A, is to chop up your RNA. Right? So this gives a little more leeway in terms of using degraded RNA. There is a risk that if it was degraded, that some RNA just might not be accessible, might be degraded too much, and you might be biasing your reaction if you really have really poor quality RNA to start. But in general, RIN actually isn't as great a predictor of the quality going into sequencing as originally thought, because you chop up your RNA anyway. So you don't want to go like crazy low in your RIN, but we actually, our cutoff is 4. So we'll go as low as 4. And one of the reasons is we're dealing with spaceflight samples too. And so when you have samples that were on the ISS, samples, tissues, let's say, that were dissected out by astronauts and then kind of frozen on the space station, and then they come back down to Earth, and then we then isolate RNA after they're back down on Earth, or a lot of times even, so anything that's not dissected on the ISS for ISS terminal studies, and this is in the context of rodents, we actually have the astronauts wrap up our carcasses, our animal carcasses, and then send down the frozen carcasses. And then we will actually thaw those carcasses and dissect out all the other tissues and then extract RNA from there. So you can imagine going through these freeze-thaw cycles, your RNA gets more and more degraded as you do that. And so oftentimes when we have our spaceflight samples, we can have some pretty degraded RNA. That said, this is another reason that we go with ribodepletion, because if you do have really degraded RNA and you are going with your poly A selection method, you need that poly A to be intact, and you need that poly A to still be attached to the messenger RNA, right? So messenger RNA is the stuff that you're interested in. The poly A is just the tool you're using to get to that mRNA. But if you have a really degraded sample and you do the poly A method, you might be grabbing poly A's that have been degraded, so that way they're no longer latched onto the bulk of the messenger RNA that you're curious about. So that's another reason that if you are working with degraded samples, that you would want to go with the ribodeplete method over the poly A selection method.
So humor me, for differential gene expression, does this then make it harder to establish differential gene expression? Because the degradation will, I imagine, other things being equal, random. So that, maybe I'm not thinking about this properly, that it's going to be harder to detect changes if it's degraded. Would that be reasonable, or is that faulty thinking?
It depends on how degraded and how much. So if the RNA is degraded, meaning that it's like chopped up, but it's still there, it still exists, it didn't go away, or it didn't get degraded so much that it's like, full of base pairs, then it shouldn't make too much of a difference. And guys, I'm such a picture person, and I have a bunch of...
So you basically, the degradation chopped it up for you, and it doesn't really matter if you're not doing poly A.
Right, so let me just... Let's see if I could do this pretty quickly here. Okay, this thick. Okay, so let's say we have our mRNA, and then I'm just going to latch onto it here, a poly A tail. Let me color that something different. Okay, so we have our RNA here, right? Our nice intact mRNA with our poly A tail, right? When we do poly A selection, we are going to pull out anything that has this poly A tail and of course anything that's attached with it. So if we have nice intact samples, intact RNA, when we pull out this poly A tail, we're going to get this whole RNA, mRNA with it, right? And so that's going to move on through the library preparation. We are eventually going to chop it after that, but this initial extraction step, that's the RNA that we're now working with moving forward. So now, if we have that same RNA, but it's in a super degraded sample, so if we take this, so now we have that same RNA, but it's super degraded, so it's chopped up. Like this, and like let's say that part of it is attached. Now when we do that first selection and we select for this poly A tail, instead of the whole message RNA coming into the subsequent steps of our process, we're now only grabbing that amount of the message RNA when we positively select with the poly A tail. So that means that we just excluded all of this from our sample and now it's going to become biased, right? However, if we do the ribodeplete method, we don't care if it has a poly A tail or not, because all that we're doing in that first step is just removing ribosomal RNA, right? So these, neither one of those are ribosomal RNA, so neither one of those are going to get removed. And so all of this is going to go into our next step, which is chopping up anyways. So this will eventually get chopped anyways. And because we didn't positively select for poly A tail, we just kept everything that wasn't wasn't ribosomal RNA. All of that mRNA, even though it's degraded, is still going to be carried out with us in the library preparation, and we're going to chop this anyway. So even this intact one is going to get chopped before we do the sequencing. Does that make sense?
It does. It does. I'm still wondering how it could introduce bias if it's more degraded. I'm thinking I have to think about exactly what's happening in the chemistry. But thank you. It makes it, I can see why the ribodepletion is important, but I still wonder whether the degradation doesn't introduce more kind of noise. It'll be harder to detect, but maybe not.
So to answer that question, you are right. So remember when we are doing our digestion, we are timing that digestion based on how large we want our inserts, right? And the size of our insert is going to be dependent on the length of the sequencing that we're doing, right? So let's say we're doing paired N150 like we do at GeneLab. So we ideally want our inserts to be roughly 250 to 300 base pairs at the end, right? So if we're starting with super intact RNA, and we do our chopping for a certain period of time because we've tested it and we know with intact RNA, we do the digestion for, X amount of time to get the size, the 250 to 300 base pair size, right? So let's say we work that out for intact RNA. Now we have a sample and it's lower, the RIN value is lower, so we know it's more degraded. Now the time that we just sorted out with our intact RNA for how long we should do our digestion for, it's going to be different for the degraded RNA. So the degraded RNA, if we digest for the same amount of time that we did for the intact RNA, we're going to take these already small pieces and make them even smaller. And so then when we do a size selection, those are going to get completely removed. Because we removed anything that's really small because we want to remove the primer dimers, right? So we'll remove anything that's like less than around, I think, 200 base pairs. And so that's where it gets tricky with how long do you do the digestion for. And so you really need to work out that digestion period based on how intact your RNA is at the start. Because if you already start with degraded RNA and you do the digestion for the same amount of time as you would intact RNA, you're going to chop up your already small pieces even smaller, and now it won't pass the size selection at towards the end of library prep.
Wow. Okay. Those spaceflight samples, really hard to work with.
They are. We have an awesome
and often unreplicated. When you look at GeneLab, and we've curated it, I had undergrads do it, there's very little replication in there that would meet the standards of what most people would use in a lab today.
Yeah, yeah. Our RIN number is 4. That's our cutoff. So as long as the RIN is 4 or higher, we move forward with it and we modify our digestion accordingly. Other questions about library prep or library QC before I harp down on this primer dimer issue?
Hey, Amanda, I have a question.
Sure.
So you talk about RNA need to be chopped. So then that means in the sequencing, you have something come from the 5' end in the middle or the 3' end.
Correct.
So at the end, how do you put together the data? Do you normalize it or you consider each segment separately?
Wyjan, you're jumping way ahead.
Sorry.
We will get into that. So
Okay.
with doing RNAseq, thankfully, a lot of the model organisms that we all work with, the genomes have been sequenced. And so we do have a reference genome available, kind of like a consensus reference genome in a variety of different databases, which we'll talk about later in the lecture. And so when we have these short read sequences, right, at the end of the day, so our libraries here, these short read sequences, how we identify where they came from is we map them back down to the reference genome. And so we can see where their complement is, where they map to on the reference genome. And then based on the region they map to on the reference genome, since we know the reference genome, we'll know where that region is that they map to. And then if that's a region that's annotated, we'll know which gene it came from, and we'll know where, towards the 5' end, the 3' end, in the middle, that it came from.
Okay, thanks. That makes sense.
Sure, yeah. We'll get into that a lot more when we get to the alignment step of our data analysis. So that is something that we're going to be doing in the course. Other questions? Oh, thank you, Ty. Appreciate that. Okay, so before we go on our actual break, which is at 1:30, sorry I was kicking you guys out early, I do want to harp down on adapter dimers. So I've alluded to this. I told you what a problem they are. A lot of you guys figured out that, you know, if we have a ton of adapter dimers in there, we're going to be sequencing adapter dimers, and that's a waste of money. Of course, it doesn't contain our sequence of interest. Even more terrible is that the sequencing, when we go and introduce our libraries to our flow cell, they're going to be latching on based on the complementary of their P5 and P7 regions of the adapter. So there's oligos on the flow cell that are complementary to the P5 and P7 region, which allows, when you introduce your libraries to your flow cell, allows them to connect onto the flow cell. And if you can imagine, if the sequence that you're introducing to the flow cell is smaller, it's going to have an easier time latching on to those short oligos, right? So you're not going to have a bunch of additional things that don't latch on kind of flapping out there and making it difficult to find and latch on. That means that even a small amount of primer dimer contamination will account for a lot more of your overall reads that you're going to get because of that bias, because there's a bias towards those shorter fragments. That's also why when you're doing your size selection and doing your digestion, you want your library fragments to be as similar in size as you can. So within reason, there's always going to be a range. So and this is just a reminder that when you assess your library quality, you're looking for adapter dimers and your fragment size, so how big your fragments are. And that's determined by, we also look at the quality of the library. So in addition to doing the RIN at the beginning, as Elva pointed out, we also assess the quality of the final libraries using a qubit or qPCR. And this is what would be 0% adapter dimer. I know we're at 1:30. I just want to get through the slide, and then I'll have you guys all go on your break. This is what a 5% adapter dimer. This is taken by looking at the area under these curves. This is your lower base pair marker, and this is your heavier base pair marker here. And so this, right, we have our libraries are, in this case, around the 5,600 range. And here we have no adapter dimer, right? Adapter dimers are going to be around that 150 range, right? Because it's roughly 75 base pairs of the P5, roughly 75 base pairs of the P7. So when those come together, you're going to get roughly 150 base pair for your primer dimer. And so this would represent 5% of your primer dimer, again, area under these curves. Now, when you sequence, if you have 1% adapter dimers, you're going to end up with about 6% of your sequencing reads are going to be adapters. Not too bad. That means about 94% are going to be your sequences of interest. So that's not too bad. So this is what we shoot for, less than 1% adapter dimer contamination. If you have as little as 5% adapter dimer in your library, it will account for 60% of your reads. That's what a problem this is. Just 5% contamination of adapter dimers will end up being 60% of your reads, which means you have to throw 60% of your reads away. If you have 10% adapter dimer, it is 84% of your reads. So you almost have to throw away everything. So again, can't harp enough. We really want to make sure that those primer dimers are gone when we do our QC of our libraries before we go into sequencing. I'm going to end there. I'm 2 minutes into our break. I'm sorry about going a little over, but when we get back, I can answer any more questions about what we just went over, and we'll carry on with the lecture.